Yeah. Yeah. Can you see?
Yes, ma'am.
Yeah. So another definition is biotechnology is the application of scientific and engineering principles of the processing of materials by biological agents to provide goods and services. Okay. So that means whatever the way they have defined, it is nothing but using of living organisms and manipulating the things, okay, for the use, for the benefit of human beings. So that is the final definition of the biotechnology.
Yes, ma'am.
So if we look into the history, so the biotechnology has origin many years ago. Okay, it is, we don't know exactly, the branch of science was not there, but the techniques were already there, because I told you yeast in the alcohol production or bread production, then curd production. Every day we, from generations, we have been doing all these things. So since the human civilization, it was going on, but as a science, only less than 100 years old. Okay. So here, fermentation is the oldest biotechnological process discovered by the people by soaking the grains and food juices. So we all know the fermented grape fermentation. So from where the wine will be produced.
And for dosa batter also, ma'am.
Yes, that is also a type of fermentation. Idli, dosha. So all these like fermentation process we have been doing since many years. And other early processes practiced by the man are bread production, alcohol beverages, vinegar, curd. Okay, so all this. But nowadays, what are the main applications of biotechnology are recombinant DNA technology. So it is nothing but we are inserting a gene of interest into the vector, that is plasmid. There are many vectors like BAC, YAC. So based on the size of the DNA fragment, size of the gene we are in, whether it is a single gene or complete genome. So based on that, we will be using different vectors. Okay, we will look into them in detail, but that technique is called as recombinant DNA technology. We are recombining the DNA, okay, 2 copies. And another is PCR, that is polymerase chain reaction. So this is a technique where we are amplifying the small amount of DNA. So usually in crime sites, okay, we will get only a small amount of DNA. We don't get the enough sample, right? We may find a single hair follicle.
In forensic.
Yes. Yes. So we may get a single drop of blood like that. So even with the small amount of DNA or small amount of sample we are getting, we will extract the DNA and we will amplify the DNA so that we can get the conclusion out of that. Even in the parental like issues, paternal identifications, all this. So many applications are there. Then cell culture and fusion. So if we want to culture the cells, okay, because before developing any vaccine or drug, first thing is we have to perform them on the cell culture. So how that particular drug or vaccine is behaving in a single cell. So what is the mechanism behind that? How it is going into the cell, how it is doing its action. That is first step. Once we know everything, the mechanism, then you cannot just go by that data, because in our body, we have all the cells which are functioning coordinatedly. It is not that each cell is behaving differently. So it is nothing but the body is behaving as a whole. It is a single entity. So every cell is influencing the other cell. Okay, the behavior of the adjacent cell or like that, even hormones, by production of hormones or cell signaling, all these things. So once we get the data from a single cell, then they will go for the animal studies in mice or any animal model. They will select suitable animal model and they will perform the experiment. So once it is successful in the animal, then they will go for the clinical trials in the human beings. Then they will release the data, like a particular vaccine or drug into the market. So here for that, first we have to culture the cells. So that cell culturing, fusion, all this comes under biotechnology application. Then gene cloning. So the same way, if we want to see how this gene is expressing, we will isolate that gene and we will clone that into another cell. And we will, cloning. Then we will try to express the gene. So how it is behaving and under some condition. So that if we want to test that thing, we will use this technique. Then another is DNA.
For example, a sheep called Dolly.
Yeah. So there are many techniques. Yeah, even Dolly, that is a kind of cloning. And even another thing is, if you want to see only the expression of a single gene, okay, so how it is doing? Suppose under the absence of oxygen, how this gene is functioning, or in the presence of oxygen, how it is doing. If you want to do such kind of experiment, so only you isolate the gene and you will try to express. That is also cloning. Okay. It is only the gene we are looking.
Okay.
Then DNA fingerprinting. Fingerprinting means we are isolating the DNA, we are fragmenting, and we want to know the structure of the DNA. Like if suppose genome is there, so we want to match bits and pieces like puzzles. How we match? How we suppose you have identified some new bacteria. You don't know, like you have broken that into small pieces. You don't know which piece comes first and next which piece will come. So for that you have to do some matching and mismatching, so that you will know what is the correct sequence of the genome. So such kind of things we can do with this DNA fingerprinting. Then environmental engineering. So environmental engineering means you are trying to use living organisms for the betterment of the environment, like to reduce the pollutants, to clean up the water bodies, surroundings. Because manually, if you want to do it, sometimes it is not possible. Like if oil spills in the oceans, so usually this oil, petrol, all these things are transported from one continent to another continent through ships. Sometimes by accident, that will be spilled over the oceans or large water bodies. Then what happens is oil will form a layer on the water body. And when this forms a layer, it cannot be cleaned. Because of that, it cannot be, so it is not soluble in the water. Therefore, that will be like permanent thing which will be there. To remove that, we need to lit it up. Okay, we want to, suppose we lit the fire, so it will get evaporated. But because of that, the
water will be polluted.
Water will be polluted. The aquatic animals and plants which are present, what temperature of the water will increase and they will die.
Exactly.
So all these problems are there. So for that purpose, without doing all those things, we can use some oil-eating bacteria, and we can just spill over this oil so that it will eat up all the oil. So it is better, like without killing anything. So like that.
In the biological way.
Yes. So like as I said, every organism has some potential, some unique quality. So we should identify that unique quality and we should exploit that.
Exactly.
So that is what we are doing. And next is immunology. So for immunology, we have lots and lots of applications. Like if we see nowadays, like immune system is very, very important to combat any disease. So there are many components of immune system are there, like cells, tissues, everything. We have antibodies, all this. So for monoclonal antibody production, everything, it includes this biotechnology field. So like that, if we look into the biotechnology, we have lots and lots of applications. So these are only some important applications.
Okay. Using biotechnology.
Yes. Okay. So history we will look into, like 6000 BC, Sumerians and Babylonians, they knew this technique of preparing beer. Okay, so as I said, it is not a science at that point of time, but it is a technique. Then
Yeah.
2500 BC, Aryans, they have started preparing the curd.
Curd.
Then 2000 BC, Greeks, they prepared cheese from the milk. Okay, it's the first fermented food, cheese.
Yes, ma'am. Yes.
Then 1822 to 1895, actually during the time of Louis Pasteur, there were many discoveries that has happened. And in immunology, especially immunology, then Koch, there are many scientists who have made a good contribution to the immunology and biotechnology. So Louis Pasteur is called as the father of microbiology and father of industrial biotechnology, because he actually gave a scientific this thing to the fermentation. Okay, so he discovered that yeast produced alcohol in the wine. Till that time, we don't know why it is happening. It is happening, that's it. But what is the process and how it is happening, we don't know. So actually, he was the one who actually gave the mechanism and what is happening behind the reason he has explained. And rod-shaped bacteria, it is producing acetic acid, which is causing the souring of wine. So these are the things he has actually explained. Then in 1929, Alexander Fleming discovered antibiotic penicillin. Okay, accidentally he discovered penicillin. Then in 1973, first successful genetic engineering experiments were carried out in world laboratories. So this is a brief history, okay, the timeline, how it has happened. So what is the scope of the biotechnology? So it is a multidisciplinary. So if you see biotechnology, you have to study biochemistry, microbiology, genetics, molecular biology, immunology, everything you have to study, because you are involving everything. It is not a separate, suppose if you want to study biochemistry, means you will study only biochemistry. What are the pathways, all these things, metabolism. But in biotechnology, it is not that thing. We are manipulating something, means we should know each and everything. So it's a multidisciplinary subject. So that has a demand in the industry. Okay. And it is a branch of advanced biological sciences. So now multinational companies are more concerned about production of pharmaceutical products like antibiotics, vaccines, drugs, gene therapy. So gene therapy also, it has gained a lot of importance. Of course, still it is not completely into the market, but slowly in Western countries, slowly the therapy has started. We have got the permissions for some kind of gene therapies. Then improvement of clinical testing, then diagnostic tools, then especially in countries like India, where it is agriculture based, we want more varieties of crops, okay, and animals, farm animals, because land is reducing nowadays. So most of the agricultural land is getting converted to industries or residential areas. So within the small area, we need more yield, and even the population is increasing. So we need the like the demand and supply is also more. We need more amount of food in the less amount of area. So for that, we need this biotechnology so that we can increase the production of the crops. Then for the food products like fertilizers, pesticides, and beverages, and some industrial, not only this is for the, okay, some we also require some commercial things like alcoholic beverages. Then health sector requires antibiotics, then amino acids, enzymes, vaccines, steroids, vitamins, insulin, human growth hormones, okay, citric acid, variety of seeds. So all these comes from this biotechnology. This is the scope of biotechnology. And also apart from this, if we see in the environmental perspective, waste treatment, okay, so the large dumping yards will have a bulk of waste, so that has to be treated properly. Then bioremediation, by using living organisms, we are removing all this waste. And energy production, because we need now petrol, coal are getting exhausted. Okay, so we need alternative sources of energy. So for that also, like biogas production, all this comes from this biotechnology. Then enzymes, DNA, RNA, like these are all reagents. When we do some experiment, we require some enzymes, and we require some DNA, RNA. So for that also, we require this. Then many biological products all over the world, for the large scale production, we require this biotechnology. So what is the infrastructure in India? Is we have 40 national research institutes, 300 life science colleges. Of course, this might have got increased more also. Then 120 medical colleges, 200 biotech companies, 100 teaching hospitals, then 15,000 scientists, 5 lakh graduates per annum are coming out, they are graduating, then 17,500 medical graduates, then 25,000 scientists in the R&D, and medical and paramedical professionals. So these many people are actually into the biotechnology field. Okay, so far in the industry, so what is the progress that have made in the biotechnology field means there are more than 370 drug products and vaccines are currently under the clinical trials for more than 200 diseases like cancers, Alzheimer's disease, heart disease, diabetes, multiple sclerosis, AIDS, and arthritis. Okay, they are still under clinical trials. Some might have already passed also, but this is overall in the world. In some countries they may be accepting, in some countries still it is not into the market. And some genetically modified organisms or crops like papaya, soya beans, corn. You might have heard BT corn, BT cotton, BT brinjal. So all these are genetically modified organisms. Then environmental biotechnology, it is to clean up the hazardous waste products. Okay, that means by using the pollution eating microorganisms without using any chemicals, we are cleaning up the waste in the environment.
Scavengers.
Yes. Yeah. Then DNA fingerprinting. So it is for criminal investigation, that is PCR, what we do, and the forensic medicine. Okay, so all these are biotechnology products. Then 1,466 biotechnology companies are there in the United States. And in that 318, only in the United States, you can see those many companies are there. And in that 318 were publicly held. And in US, 1,94,600 people are employed by 2002. Now it may got up like much more. So like that, if you see, no need to remember all these numbers, but this is to say like what is the scope of biotechnology. Okay, so here in US, so this is mainly regulated by FDA, Food and Drug Administration, and Environmental Protection Agency, and Department of Agriculture. So please mute your microphone. So in the same way, in India also we have Department of Biotechnology, Department of Science and Technology. Like that we have CSIR, ICMR.
Data.
Please mute yourself. So in India also we have many agencies which are giving the fund and to encourage the research on this biotechnology, various aspects of biotechnology. So research is conducted by small companies, large corporations, and public universities. If we see now, it is not that only we do biotechnology research in industries, but we can in startups. Small companies means we have startups, so where we can take only one objective and we can do research, and even large corporations like Biocon, all these are biotechnology, Monsanto. So we have some biotechnology companies. Then even universities. We have Department of Biotechnology in every university where we perform some biotechnology research. So funding comes from public and private sources. So public means government. So as I said, Department of Biotechnology, DBT is there. Then Department of Science and Technology, then ICMR, CSIR, Scientific Council for Research, Industry and Research. So like that, we have some government agencies which are providing the funding. Not only that, we have some private companies and foundations. Okay, so even they will, like Tata Foundation or Bill Gates Foundation, like that, even they will give the grants to perform the research on the biotechnology aspect. So in the California, they have allocated $300 billion for the research of stem cells. Only to perform the research on the stem cells, they have given that much fund. So this is how the funding is coming. The main focus of biotechnology is on health and medicine. Okay, because even now, especially after COVID, it has become a need of our okay, because we require a lot of vaccines. Healthcare sector should be strong. Okay, so we might get lots of pandemics like that maybe in future. It is a sudden, we didn't expect this to happen. So that means the sector, healthcare sector and medical sector should be very stringent. Okay, so the main focus is on the health and medicine. Then food science, as I said, because we have the demand is more and supply is less. Food supply, so we need to increase the food supply more. Then environmental science, that means to clean up the environment. So because of pollution and all these things, lot of problems there which are directly targeting the human beings. So to reduce that, again, we require this kind of science. And even agriculture, agri-science. So genetically modified organisms. So here it is a general term. Genetically modified organism means it includes animals and plants. If we specifically has to say, we have to call them genetically modified plants or genetically modified animals. So it is consumed by millions of people every day in America, especially. So we have the best examples are BT cotton, BT brinjal, BT corn. Okay, then tomatoes, flavor saver tomato, then papaya. So to increase the shelf life for something and to
golden rice also.
Yeah, golden rice. Yeah, that's correct. So golden rice. So you have lots of varieties. So each one has golden rice is for improvement of nutrition. In the same way, this flavor saver tomato is for the increase the shelf life. Then BT crops are to protect the crop from pests. Okay, like that. So we have many modifications in the crops. And almost 56% of all soybean plantings in the worldwide are genetically engineered. And especially in the US, the use of these genetically modified organisms are more. So in US and in other countries, like in 18 countries, 167 million acres were used to produce the genetically modified crops in 2003. Okay, but now still it might have got increased. But there are the negative aspect is also there. A lot of protest is also going on from the another set of people. Okay, some set of people, they are consuming, and another set of people, they are objecting. So both are there, but still it is under the use. So the NC Biotechnology Center, what it predicted is that biotechnology industry in the state will contribute more than $25 billion for the annual income each year within the next 25 years. This is in the US. Okay, so according to the experts, That means there is a lot of scope for the biotechnology industry. So pioneers of biotechnology. So they have not actually done something like biotechnology, but they have they have invented something. They have done research on some basic aspects on which the branch of science biotechnology was developed actually. Okay, so Antony von Leeuwenhoek, he discovered microscope in 1675. Okay, actually he was a merchant to look the holes on the cloths. So actually he invented something called microscope. But later, due to his interest, he took water, rainwater, and he just saw under the microscope. He could see some small animal kind of things which are moving. He called them as animalcules, like small animals. But later, after many research, they were like bacteria. Okay, so which are moving. So that means knowingly or unknowingly, they are the ones who have laid the foundation for the basics of biotechnology. The next is Girigor Jahan Mendel. So everybody, science students, they will know Mendel. So in 1863, he was an Austrian monk. He first conducted genetic experiments in the pea plant. So he, in his own garden, he has selected garden pea here, and within garden pea, he selected 7 qualities, 7 traits, which are contrasting. That means they are only 0 or 1, like that. There are no shades. So like if it is tall or short, only 2 phenotypes are possible in each trait. Okay, so like that he has selected and he has done some experiments. He published his results, but at that time nobody accepted what he has done. But again, after 100 years, okay, there was no genetics, nothing at that time. So this was in 1800s. But after 100 years, after his death, so another scientist, they have read his work and they have reconducted the same experiment. Then they got the same result. By that time, the science was advanced and they could actually get some inheritance, how the pattern of inheritance is happening in the human beings or plants or animals. So therefore, the branch of genetics was developed and Mendel was called as a founder of genetics. Then Louis Pasteur. So in 1970s, he has done lots of discoveries. So some of them are, he disproved the notion of spontaneous generation. So at his time, there was theories like how the life has evolved. So there was a theory called spontaneous generation. That means spontaneously the life has evolved. But they could not prove it. How spontaneously it will evolve. So there should be some background for that. So actually he was the one who disproved that by performing some experiments. And he also discovered the role of bacteria in spoilage. How this bacteria is actually doing some like due to some mechanisms, it is spoiling the food. Yeah.
Contamination.
Yes, contamination. Even in the food, yeah, contamination the same way. So it is by releasing some enzymes. Okay, it is spoiling the food. So how to prevent, we have to add salt, all this. That is another branch again. So actually he described the role of the bacteria and also scientific basis for the fermentation. So we have discussed already. And he created the rabies vaccine.
Dog bite.
Yes. Then Robert Hooke, he was the one actually to discover the cell itself. Okay, so that means because these are all basics. Okay, this is the basic for like Louis Pasteur, whatever he has discovered. Like if Antony von Leeuwenhoek would not have discovered the microscope, so there was no science was there, because the other person might, maybe he will discover the microscope later. Okay, he will invent the microscope, but the science was developed because there were some equipment, some techniques which were given by these scientists. Okay, they have not completely developed a branch of science, but these are the bits and pieces from where this different sciences have evolved, like cell biology, molecular biology, biochemistry, microbiology, biotechnology, all these things. So Robert Hooke, he was the one who discovered the cell. So from that only we now got the cell biology. So in 1665, he invented the compound light microscope and he observed the cork, wooden cork. And he could see that some compartments, some room-like structures are there. So then he observed and he named them. Okay, so later this was discovered to be the cell of the plant. Okay, they are the compartments are the plant cells. So first he observed the cells. Then these scientists are very like, they have changed the complete science. James Watson and Francis Crick. So by the discovery of DNA, okay, by giving the structure of the DNA, so the complete science was changed to the next level. So in 1953, Englishmen like Watson and Crick, they are responsible for the discovery of double helix structure of DNA by using X-ray photographs. So before them, Rosalind Franklin, she has done most of the data, everything. She was the lady who worked on the structure of DNA, but before presenting, she died. So that based on that findings, these scientists, they have, and these people also, they have done the experiments, but she has given the additional information. So by clubbing both, they could come to a conclusion about the structure of the DNA.
What's her name, ma'am? Name?
Rosalind Franklin.
Okay.
Okay. Yeah. Then next is Paul Burke. In 1972, he was from Stanford University. He's a scientist, and he first developed recombinant DNA technology. So from this time onwards, the biotechnology, actual biotechnology was started. It is the method for insertion of genetic material from one organism into the another. Okay, rDNA technology, what we call. So historical development. So in 1750, the same thing, it repeats mostly. It is like cheese production, yogurt, bread, which we have already discussed. Then in 1869, DNA, it was discovered in trot sperm by German Meister. So even like this is like DNA structure was given by Watson and Crick, but by that time the DNA was known. But there were another scientists who have worked to tell that DNA is the genetic material. So till that time, there was DNA, RNA, and protein. But scientists were confused which is the genetic material, whether it is DNA or RNA or protein. So there were scientists, there are a lot of experiments. Now no need to discuss all these things, but they have actually given that DNA is the genetic material. Then this Watson and Crick, they have given the structure of the DNA. Okay. So now this in 1869 itself, the DNA actually was discovered in the sperm cells. Okay. Then in 1919, a Hungarian agricultural engineer, he used biotechnology term for the first time. So in 1940s and 50s, the structure and function of DNA was actually investigated. Okay, once we got the structure like by Watson and Crick, always remember, structure will tell you the function. Okay, so especially the DNA, the sequence will give you the structure and structure will tell you the function. If we know the sequence, we will know like where it is, how it is folding, even in the proteins. If we know the sequence, we will know the structure. We can get the structure out of that. Once we know the structure, we will know the function. So in the same way, from 1940 to 1950, the complete research was into the structure and function of the DNA. And in the DNA also, we have different forms. It is not that a single form. We have A form of DNA, B form of DNA, Z form. Like that, we have many forms. And we have, it is not only the double helix. In some animals, it is single helix. In some organisms, not animals, a single strand, double strand, circular, linear. We have triplicates, quadruplets. Like that, we have many forms of the DNA. So in 1980, US Supreme Court, it approved the patenting of genetically altered organisms. So till that time, there were no patenting all this, but now it has increased, like for intellectual property rights. The patenting techniques were increased. Then in 1980s to 1990s, a variety of genetically modified organisms by using biotechnology techniques was increased, and that were used in agriculture to medicine. Everywhere they have used this. So for this, they have used recombinant DNA technology. That means to extract the DNA. Suppose golden rice. So what is this golden rice? In the rice, we are introducing the beta carotene gene. Right? So beta carotene is not actually present in the rice. It is coming from somewhere. So we are isolating the beta carotene gene from some other organism, and now we are introducing that gene into the rice crop, so that whatever the rice we consume, we will also get the vitamin A. So to improve the quality and nutritional value of the rice. So this is how we are introducing the genes or genetic material from one organism, and we are introducing that into the another organism. That may be plant or animal. And along with this plant tissue culture. So that means we are culturing the plants which are genetically identical. In a small space, we can culture many plants. Okay, we can grow many plants which are genetically identical, and we might have the plants will have some quality which we require, like secondary metabolite production. Okay, like that we will discuss other examples. So this is plant tissue culture, and this is from the explant. Explant is nothing but we are taking some parent plant from where we are taking some part of the plant, and we are culturing that part into many plantlets, small plants. So the plant part from where these plantlets are coming, that is called as explant. Okay. So in 1990s, the first transgenic organism was actually introduced in the agriculture production, like BT corn and soybean. BT means Bacillus thuringiensis. So this is a natural insect resistance for these crops. So how this is happening means from Bacillus thuringiensis, it's a bacteria. It has a gene which will produce a toxin. So they have isolated this gene which is produced. It is protoxin. That means it is inactive in that particular stage. So that particular gene is introduced into the crop, whether it is corn, brinjal, cotton, or soybean, or whatever. Whenever an insect comes and eats that particular leaf or some part, this gene enters into the, or this protoxin enters into the insect. So when this protoxin enters into the insect, so within the gut will have the pH of acidic pH, right? So during the acidic pH, this protoxin will become active, and it will become the active toxin. When this toxin become active, that will start functioning on the gut of the insect, and slowly the insect will die after ingestion of this toxin. Okay, this is a mechanism. So this was first actually introduced in the 1990s, but there was a lot of protest from the some sector of people, because if we eat, or the animals, if they eat this gene, that may affect. But mostly what the scientists say is that it is not effective in, it is not causing any side effects in human beings and animals. It is only causing death in the insect. But still there are some protests which are against this. Then in 1997, Dolly, this was the first animal which was cloned from diploid cells produced in the Scotland. So this is from the, like they have used a somatic cell. Okay, usually germ cells, the organisms develop from the germ cells. From whenever the fertilization happens from the egg and sperm fusion, then the zygote will form, and then the organism will be developed. But here they have used somatic cell. Okay, so that means by manipulating the somatic cell also, they have produced the organism. Of course, it has died after some period of time, but they were successful. Then in 1990s, late 1990s and early 2000s, human cloning is outlawed in the US, and the first concerns over the use of human stem cells in research begin to arise. So, as expected, once the science is developed, it is with a good intention. But after some years, what happens? That will go into some other aspect, and it will be completely for the, it will go beyond certain level. So where it has to be stopped. Okay, so this human stem cells. So stem cells actually were discovered during this Hiroshima and Nagasaki attack. So accidentally they have discovered, like after this attack, some generations of people who are living in that period, they have some blood related diseases. Okay, one, the generation is okay because it is a nuclear atom bomb. So okay, that generation is affected, maybe next generation, like that some 2 to 3 generations are expected. But still many generations, they are having the same kind of diseases. So then the scientists have looked into what is the reason for that, and they could discover that there are some cells which are present in the bone marrow, which are called stem cells. So these stem cells, they have the potential to give rise to any type of the cell. So actually we have different types of stem cells. During the embryonic stage, whatever the cells which are present, all of them will be having the potential to give rise the complete organism, because after fertilization, the zygote, which is a single cell, that will be formed. From a single cell, now we are getting these many cells, and complete organism is developed. That means the zygote has the potential to form a complete organism. In the same way, there are some cells called stem cells which can give rise to any type of body part. So because of the mutations in the stem cells, the people who are in the area of Hiroshima and Nagasaki, they are getting the same kind of diseases. That's what they have discovered. So after that, they could identify the potential of these stem cells, and they have started doing the research on the stem cells. Okay, nice that scientists have developed many, that we can use the stem cells to cure many diseases. That's great. Then they have said that embryonic cells, those cells which are there in the embryo, they have the capability to grow into any type of cell. Then what happened in US, the mothers, they have started to abort their embryos to donate for the stem cell research, just for the sake of money. So that has increased. So now we have surrogacy. So how we have got restrictions on surrogacy, in the same way, they have started to just give these embryos for the research. So it has become like a business. So then again, the law in the US has stated, they have given some regulations on this stem cell research, and they have some period of time, I think they have banned also, but they have imposed some regulations on this stem cell research. Then the agriculture field, different agriculture areas like plant science, animal science, environmental science. So all this we have already discussed the focus. Then in the plant sciences, especially transgenic plants. Okay, so the plants which are having another quality from another plant. So that is called transgenic plant. So those, the increase, the research is now focused on this transgenic plants to get more amount of products from the less amount of plants. So here how it is done, you can see the picture here, DNA with the desired gene. So if suppose we want some gene like beta carotene, so we have isolated that gene. Now what we will do, the DNA, here we need some vector. So like if for insulin production, we have used the vector plasmid from the E. coli. Here we are using agrobacterium. So agrobacterium is a bacteria. It will usually infect the wounds, okay, in the plants. So what the scientists they have exploited is that first we have to take the isolate the DNA. We suppose I want to produce a golden rice or like that. Okay, for example, golden rice. So what I will do first, I have to isolate the beta carotene gene, and I will take some metal particles like gold or silver, which is suitable. So that I will coat with the DNA and I will keep it ready. Then in the agrobacterium, we have two types of agrobacterium. Agrobacterium tumefaciens, which will have Ti plasmid, and agrobacterium rhizogenus, which have Ri plasmid. So rhizogenus is used for the hairy roots in the plant.
Ma'am, can you please repeat the points?
Yeah. So first thing is, first I have to keep the ready. I have to keep the gene ready. Okay, whichever the gene I want to insert. Like for example, I'm telling it, not that in the golden rice they have used this technique. I'm just giving you the example. Suppose I wanted to insert beta carotene into the golden rice. So first I will select a plant in which, like carrot, which will have the beta carotene. First I will isolate that gene and I will coat the metal ions, because directly you cannot keep the DNA. It is not visible for you, and it requires some carrier, because the DNA can be easily degraded if the temperature is raised, or if pH is changed, or if DNA is enzymes are there. So like that, many possibilities are there. So because of that, we have to make some support. So we can use the metal ions, so metals like gold, silver, which are not reacting with the DNA. They are only acting as a carrier for the DNA. So I'm taking some metal and I'm coating that metal with the DNA and I'm keeping them aside. Then I want to insert this DNA into rice plant. So before that, what I have to do is I have to make the agrobacterium, which is a vector, which will carry this DNA, foreign DNA, into the rice plant. So for that, what I am doing is I am taking the agrobacterium bacteria. So agrobacterium, it is two types. Agrobacterium tumefaciens. This is one type of bacteria that is having a plasmid which is called as Ti plasmid. And another one is agrobacterium rhizogenus, which is having Ri plasmid. So you have to select which bacteria have to be used in this experiment, because Ti plasmid is used to insert the DNA into the shoot part, whereas Ri plasmid can be inserted in the root part, right? So therefore, suppose if I want to introduce the DNA in the shoot part of the plant, I have to take the Ti plasmid of the agrobacterium And in that TI plasmid, I will have some region where I can insert this foreign DNA. So first I have to cut that region using restriction enzymes. I cannot cut with the scissors, right? It is a DNA. So we have to cut them in specific points by using restriction enzymes. Then I have to, whatever the DNA which is I have coated with the metal ions. So from that, the DNA has to be isolated, and that here two types. One thing is directly I can take the DNA without coating any metal. Okay, just DNA and inserting in the agrobacterium vector and directly injecting in the plant. That is one method. And another method is without using any agrobacterium, directly I'm taking the DNA which is coated to the metal, and there will be some guns, small guns. Okay, that is called particle gun method. So I have to insert this DNA with metal into this gun, and I have to shoot on the plant so that this metal along with the DNA will go and it will get inserted in the plant. Okay, is it clear?
Yes,
Any doubts? Okay.
It acts like a gun or means
Madam, it is nothing but a gene gun method, no ma'am?
Yeah, gene gun or particle gun. So it has many names. Okay, it is like gun is nothing but it is not a physical gun like injection. We are just injecting that. It's a process, like we are with a high force. Okay, with a high force.
Thank you, ma'am.
Yeah, we are with a high pressure. We are making this DNA with the metal to enter into the plant, and it has to go and it has to get inserted in the genome of the back plant. See, unless the foreign DNA goes and enters into the genome of the plant, that cannot go to the next generation, right? It will stop only at that generation. And it won't express, first of all. Right? Is it clear?
Yes, ma'am.
Yeah. So, either of the methods, see, based on, we have many methods, not only these two, we have 10 methods, more than 10 methods to insert a foreign gene into the, we have what you call, we can just make the pores, electroporation you say. Okay, that means we will make the pores by using the electricity. So like that we have many methods, but based on the plant and based on the gene, okay, we have to apply the technique, right? This is only for your understanding and example, just simple example. So by any method, first what you are doing is you are inserting the foreign DNA into the genome of the plant, target plant. Now you have to select, okay, first you will divide this plant into multiple copies in the tissue culture. Once you divide, first you have to select, okay, because not every plant will have the foreign gene. Okay, some maybe it will be 50-50. Some plants may have this foreign gene and some plants may not have the foreign gene. So therefore, you have to carefully select which plants are having this gene, and only that plants you have to take and you have to culture them. And then you have to, once they are grown to sufficient size in the lab, then slowly you have to transfer them into the pots in a controlled condition, like everything, temperature, humidity, light and dark period has to be controlled. So all this, once it is adjusted to the environment, then it can be introduced in the crops. That means in the field, right? So this is a simple technique, the overall technique. Again, every step you have many options. So based on the experiment, you have to choose. Okay. Now in the animal science, we have in vitro fertilization, artificial insemination. So they are nothing but the breeding programs. So in vitro fertilization, even in the human beings nowadays, we are seeing this IVF. So in vitro fertilization is nothing but we are selecting in the couples, we are selecting the sperm or semen sample from the male partner and egg from the female partner. So before that, first of all the hormonal levels, all these will be checked. If any treatment is necessary, so that will be regularized. The ovulation has to be regularized, and the sperm and this egg will be collected, and the quality will be checked. And then it is the fertilization process is done in the outside the body, like in the lab. So in vitro means in the lab, in the tube, actual meaning is. So in the lab, the fertilization is done. So once the zygote is formed and it will start dividing, 8 cell stage like that, again that is inserted back into the mother. So then they will check for the stability and the rest will be continued. So this is a technique. Then another one is, this is usually now it is doing in the human beings. Then artificial insemination is mostly done in animals where we want more number of animals. Okay, so as I said already, what is artificial insemination? Insemination is in terms nothing but the introduction of the semen sample into the female. But here we are doing it artificially. Okay, so improve the breeding techniques. Then environmental science. So I told you, like for cleaning contamination and for protecting endangered species. So nowadays, because of various factors, most animals and plants, they are getting endangered or extinct. So we want to protect the, at least we can save the gene pool, genetic information of those organisms, so that we can regenerate the population by cloning techniques. So in this way, we can protect the endangered species. And bioremediation, it is nothing but the cleaning up of the contaminants by using the organisms like bacteria. Then immunoassays, to test soil, water, blood. We all know, like ELISA, enzyme linked immunosorbent assay, then RIA, radio immunoassay. Like that, we have many immunoassays. Like we can to know the reactivity, how this antigen antibody is reacting. Okay. Then installation of biological barriers to prevent the transfer of harmful microorganisms between production facilities. Like in industries, usually to stop the growth of microorganisms. So they have been using this biotechnology methods. Okay, then pharma. So for like production of vaccines, drugs, medical substances, and also to prevent the contamination in the food sources. So then DNA analysis. I told you DNA fingerprinting and PCR techniques. So like this is for paternity testing also, even in the crimes and genetic ancestry, even not only in the crimes or like that. If you want to know the ancestry of the animals, and if you want to know, suppose you have isolated or you have identified some new species, so you want to know whether this species is somewhere related to any other bacteria or any organism, so you can just test the DNA of both and you can know how they are related. So that I told you, like if you know the sequence, you can get the structure. If you know the structure, you can predict the function. So if both of them are very closely related, they will have the same function mostly. Okay, like that we can test the genetic ancestry of animals. But there are some problems also. So what are the problems? First is transfer of genes found in the transgenic animals into the natural population. So that means we are introducing only in some portion of crops or animals, but that may get spread in the other species also, which can be, sometimes it is good, but sometimes it can go reverse also. Okay. And terminator genes can be used to minimize this risk, but again there is a problem with the terminator genes. So what are terminator genes means? In the seeds, if we have suppose BT gene, so after that crop, that gene will be terminated. So they are called terminator genes, so that it won't get transferred to the next generation or any other crops. But the problem is farmers, they don't know all these terminator genes, all these techniques. Like we don't know in the
What
Western
is
country,
terminator gene? Please repeat once again.
the genes which can terminate like after one generation. Like suppose a gene is there, like BT, bacillothuringiensis, we have some gene, BT gene. So that BT gene, it will be terminated in the same after one generation. So it won't get transferred to the next generation, right? That means the seeds in the next generation won't have BT gene. Got it?
Only once used means.
Yeah, only once used. Or it will have some lifespan, maybe one generation or two generation. After that, it won't be transferred.
Okay, fine. Thank you, ma'am.
Yeah. But the problem is, especially like in countries like India, nobody explains to the farmer that there are terminator genes. So you should not, like, we have to educate the farmers before selling the seeds. But what is happening in some regions, the farmers are buying this BT crops which are having the terminator genes. See, we all know in India, we have the tradition, like usually the seeds from one crop will be used in the next crop, right? But here they are buying by using this, the BT genes will have more amount, like the cost will be more. So they are buying, like they are thinking that, okay, this gene will be there in the every generation, but they don't know that there are terminator genes. So first of all, we need to educate them so that there are terminator genes. This is useful only one generation or two generations. Then if they are interested, they will buy. But who will educate them? Like here the system is not so good to educate them. So most in some parts, what happened is, without knowing, the farmers were into loss, because in the next generation that gene won't be present. Again, the crop will be damaged by the insect. So in that way, by paying lots of money also, there is loss. So this is some kind of internal problems. Okay. Then there are some unexpected impacts of the genetically modified organisms in the environment. Sometimes they will get accumulated in the environment. Okay. So that the insects also will become resistant after some period of time. So the resistance will be developed. And expensive also. The cost of production of transgenic animals, like transgenic fish, all these things, it is very expensive process because we all do in the lab. So therefore we will sell them also for the more cost. Okay, so all people, it should be affordable, right? Then concerns about the safety and ethics of incorporating genetically modified organisms into the food of human consumption, food for human consumption. So especially in the Western countries, now the protests are mainly about the ethics and safety. Because if these genetically modified brinjal or corn are used in the foods, see, not all people are, their immune system is not same. Right? Some people may get sensitive, or some may be allergic to some things. And if the genetically modified organisms are present, then it can cause some kind of disease in some people. So there are some ethics and safety concerns, like allergens, especially the use of starling corn in taco shells. It was not approved for the human consumption. So finally, what is the use if that is not suitable for the human beings? So it's a waste of time and waste of money doing all these things. So that's why we have to be very careful in choosing like the products, which should be ethically correct, as well as it should be safer to use. And we should not disturb the ecosystem before doing that. and lack of education. That's what I have said. Before selling the seeds or products, not only seeds, any product, both the consumer and the producer, they should know like from where this product is coming, like what are the side effects and what are the positive angles. So what are the concerns? So everything, lack of education among both consumers as well as producers also. Okay, because some they usually say, okay, BT means nice. They'll just by the name and brand they will go, but they should properly know what it is. Okay, so scientific education is also important before using the products. So then before starting the biotechnology, like we should be aware of some terminologies. Okay, so what are they? First is cloning. So cloning, as I told you, gene cloning or animal cloning. Gene cloning means we are inserting the DNA or genetic material from one organism and inserting them into the another organism. So that is gene
Anywhere
cloning.
we inserting, madam?
Yeah.
In plant, anywhere we inserting gene?
Not anywhere.
Which part, madam?
We have to use the plasmid. So like plasmids are nothing. We should have the vectors.
Oh.
So, okay, first of all, anywhere means we have, suppose we have a target organism. Suppose a plant, and you have a source gene, target gene. This gene you want to introduce in the leaf of a plant. Okay, you will have some certain area where you have to introduce, not randomly. We cannot just, suppose if you want to insert the insulin gene, we cannot just randomly insert, right? Only the pancreas that insulin gene will express, right?
Okay.
So
Okay.
in that way, we have to study first of all. That's why I am telling, when we have to do biotechnology, we have to study plants, animals, we should know biochemistry, immunology, everything. Why means you should know the target gene, what are the factors, what are these things, and also you should know the organism, like where the genes are expressed, so what are the concerns, everything. So suppose, so before these two processes, like target gene has to be inserted into the target organism. For this transportation process, we will have some vectors. Vectors are nothing but some agents which will help in the transport of gene. So they can be, these vectors are usually from bacteria. So we will have plasmids, usually used as a vectors. So what are these plasmids? Plasmids are naturally present in the bacteria, and they are extra genetic material. That means normally we have DNA. So even the same way, even the bacteria will have DNA. So apart from DNA, there are some other genetic material which are not important for the growth of the bacteria. They will be usually present in the bacteria, but they don't have any use. So what scientists have done is they have identified such plasmids. The plasmids will be usually circular, and they will also divide. Whenever the E. coli, suppose E. coli divides, DNA will divide. It will become 2 copies. In the same way, plasmid also will become 2 copies. So that means it will also, it has the self-replicating capability. And another thing is it will have a marker. So it is like some antibiotic resistance markers will be present in the plasmid. So we should know, if we have the marker only, we can identify. Suppose if we say like you go to that address, without knowing the address, how will you go, right? You need some landmarks for going somewhere. So in the same way, to identify the bacteria with the foreign gene, you should have some marker. Okay, so even these plasmids will have some markers, so that after inserting the foreign DNA, we can easily identify which bacteria is having this foreign DNA and which bacteria is not having the foreign DNA, right? So there are many characters for selecting a plasmid that we will discuss if time permits in next classes. But we have vectors, not only plasmid, we have cosmids, Then we have yeast artificial chromosomes, bacterial artificial chromosomes. Like that, we have many types of vectors. So based on, suppose you have 100 base pair of DNA, you will use one kind of vector. If you have 1000 base pairs of DNA, you will use another vector. Okay, like that, you have to select the vectors. So by this, the process of doing this is nothing but the cloning. So the same thing, if we do it in animal, that is animal cloning. Okay. Then clonal offspring. So that is what animal cloning is nothing but clonal offspring, where you are inserting the complete genome into the another cell, so the another animal, so that a complete organism is formed. Not a single gene, but a complete organism or offspring is generated. Then next is DNA, deoxyribonucleic acid, which is DNA. So we should know what is DNA structure. I hope everybody will have the basic knowledge about the structure of DNA, what is the sequence, what are the components of DNA. Then genetics. So we should know first the pattern, how the genes are transmitted from one generation to another generation. So here we have some exceptions. So Mendelian pattern of inheritance is normally seen, but even non-Mendelian pattern of inheritance is also present in some organisms. So we should know in which organism how the offsprings are generated, how the pattern of inheritance is there, and also some diseases, because autosomal dominant diseases, and some animals may have some diseases. If you do all these experiments by spending a lot of money in the diseased animal, so it's a waste of money. So first of all, we should know the genetics. Okay, we should look for all these things. Then genetic engineering. So genetic engineering is nothing but first you know the genetics. You should know everything about the gene. Then only you can engineer the gene. Engineering is nothing but the modifying. So where to cut the gene? So how you cut, I already told, by using restriction endonucleases or restriction enzymes. So you cannot randomly pick some enzyme and you cannot use in cutting the DNA. First of all, you should know the restriction sites. Restriction sites are nothing but where this particular enzyme goes and cuts the DNA. Even this enzyme, it won't go and randomly cut everywhere. It will identify some sites and it will go and cut. So first of all, we should know whether that sites are present in the DNA. And then we should choose the enzyme, which enzyme is suitable for cutting the DNA. So like that, there are many aspects before cutting the DNA, before inserting the DNA into the vector, all this. So that all comes under genetic engineering. Then genetically modified organisms. So I told you genetically modified plants or genetically modified animals. Okay. Then ribonucleic acid, that is RNA. So even RNA is also important, like because the protein is this information comes from the RNA itself, because DNA is expressed into mRNA, mRNA to protein. Then transgenic organism. So the organism which is having the foreign gene, that organism is called as transgenic organism. That gene is called as transgene, right? So these are the some technical terms which usually we use.
Ma'am, what is transgenic organism?
The organism which is having the foreign gene, like golden rice, it is having the beta carotene gene, which is not from its own. It is coming from something outside. Now this golden rice can be called as transgenic organism. Understood?
Ma'am, GMO full form, ma'am?
Genetically modified organism.
Okay.
Okay. So these are the technical terms which we usually use in the textbooks, or while I'm taking the classes also, I will use. So you can just you can ask me if you are in doubt, but
Ma'am,
yeah.
if you are doing cloning, means then after transgenic organism will comes na?
Yeah.
Fine, thank you.
Means I am telling you, like cloning, if you are doing the, like in organism, you will get the transgenic organism.
Yeah, that
Yeah,
is the...
Yes. If you are inserting a gene into some other organism. That organism now has the gene which is called transgene. Trans means transfer, right?
Okay, fine.
Yeah, so that is transgenic organism. Okay, so this is about the introduction to biotechnology. So is it clear? And if you have any questions, you can ask. Anything like related to biotechnology, if you have any doubts. Is it clear?
Yes, ma'am.
Yes,
So
ma'am.
every aspect, right? Okay, just remember this technique. Some we will repeat again in the next classes. So according to syllabus, we will go some topics we may not cover also, but these are repeated in the other, not only the animal biotechnology, even in the environmental biotechnology, marine biotechnology. So in all those things also, you will encounter the same techniques, right? So just a second. Okay. Fine. So no more doubts, right? So everybody,
Yes,
everybody has a syllabus copy.
Yeah, we have books,
Yes,
ma'am.
ma'am. We have textbook.
Textbooks. Okay, okay. So in the next class, we will look into the histology and cell culture techniques. Okay. So I'm ending this class now. If you don't have any doubts.
Second unit, ma'am.
Yeah, no, in the same unit. The next topic. Yeah, unit 2. Yeah. The block 1, unit 2. General histology and cell culture techniques.
Today first one is over now, ma'am.
Yeah, first is over. Okay, fine.
Okay, fine. Thank you, ma'am.
Yeah, thank you everyone. Thank you.
Thank you, ma'am.
Thank you.